Well, hello everybody. Very excited to get underway here with our first video lecture. We're going to build right on Mendel's basic rules and principles to talk about exceptions to those rules and principles as well. These are all things that would make Mendel say, what's going on here? But as we dig deeper and we look beyond the initial presentation of these genetic traits and their transmission, we see that they do indeed follow Mendel's rules. So let's get to it. What we're going to be talking about first today is incomplete dominance and codominance. And then we'll move on to partial or incomplete penetrance. From there, we'll go on to epistasis and complementation. These are both examples of different genes interacting with one another to control a trait. We'll go on to sex-influenced and sex-limited traits, environmental influences on phenotypes, and that'll wrap up the video for today. So before any of that, we do want to just make sure that we are comfortable with Mendel's principles, and really Mendel's overarching idea of genetics relies on this notion of dominance. So Mendel's description of dominance is very clear and quite simple. When you have two different alleles in a genotype for any one gene, in other words, when that gene is heterozygotic, one allele is stronger than the other, and whatever that stronger allele codes for is what's expressed in the phenotype. So this is how dominance works at a large number of genes. Even in our complex genome, we see classic Mendelian dominance play out this way, but not always. So, for example, blonde hair is little to no pigment in the hair, and that's what makes hair naturally blonde, is that pigment is not getting into the hollow hair shaft. So that means alleles for blonde hair are actually lack of function alleles. They're mutant alleles. Pigment's not being made. We're supposed to make pigment in our skin. We're supposed to deposit pigment in our hair. And if we're not doing that in our hair, that's a lack of function. Something went wrong with those alleles. Brown hair, on the other hand, is pigment in the hair. If we have brown hair, we're seeing brown hair because pigmentation was deposited in the hair shaft. So alleles for brown hair encode the proteins that work to deposit that pigment. So if we think of it through that lens, if we're homozygous dominant, we've got two alleles that both can tell a cell how to make and deposit pigment, right? So big B is make pigment, big B is make pigment. We're homozygous dominant. We have brown hair. The cell knows how to deposit that brown pigment into the hair. Heterozygotic, well, now we see that one of these alleles tells the cell how to make pigment, and one of the alleles doesn't, right? That recessive B allele, that's the lack of function allele. That's the allele that's mutant. That's the allele that can't deposit hair, but we still dye in the hair, but we still see brown hair in this individual because that one dominant allele still tells the cell how to make and deposit pigment. You only need one working allele in order to do this job, so we still see brown hair. In fact, the only time that we see blonde hair is if the cells don't know how to make or deposit pigment in the hair at all. So that's recessive, little b, or mutant, lack of function. The cell is naive as to how to make and deposit brown pigmentation, and so we don't see that in the hair, and the hair is blonde. So played out this way, we see Mendel's dominance, right? We see that the brown-haired allele is dominant because that's the allele that has function. That's the allele that can tell the cell what to do. And if you have two alleles telling the cell what to do, or just one allele telling the cell what to do, the cell still knows how to make and deposit brown pigment, and you'll have brown hair. It's only when the cell has no information, two recessive alleles, do we see blonde hair. So this is a functional definition of Mendelian dominance, and it's based on what alleles can tell cells what to do. But what about the case when you actually do need two working alleles to make your phenotype, to make your ending cell product? What if you need both of those alleles working together to control the trait? Then Mendel's apparent definition of dominance breaks down. If the only way we could make and deposit brown pigment was by having two working alleles, then the only genotype that would give brown hair would be homozygous dominant, for example. What about having two different alleles that each do something different? So it's not brown hair, it's brown hair, red hair, and now these two alleles are competing with one another. These alleles would fight for their expression rather than one be lack of function. It would be two different gain of function alleles. So in exceptions like that is where things get interesting, and where it appears that we break Mendel's rules, but in fact, we're not breaking Mendel's rules. We're just getting to a nuance of genetics because of the different way that these alleles behave. So with that framework in mind, let's jump into first incomplete dominance. Complete dominance, that's what we just discussed. Complete dominance is when one allele is all you need to give the dominant trait. And if you have one allele that's dominant, in other words, if you're heterozygotic, then you have the dominant phenotype. And if you have two dominant alleles, you have the same exact dominant phenotype. Brown hair, blonde hair. That's complete dominance. In other words, there's no phenotypic difference between being homozygotic dominant and heterozygotic. But there's also incomplete dominance. Incomplete dominance is where the heterozygote has its own unique phenotype. And that phenotype is usually an intermediate phenotype between the homozygotic dominant phenotype and the homozygotic recessive phenotype. So a classic example is in flowers. Maybe homozygotic dominant for R, big R, that gives you red flowers. Homozygotic recessive, little r, gives you white flowers. But in order to make that red pigmentation, you actually need both dominant alleles. If you only have one dominant allele, you make half as much of that red color, you make light red. You know, we call light red pink, but pink really is just light red, and that's the phenotype for the heterozygote. So we can see this pretty straightforwardly in plants of this kind. We can get a red flowering plant. We know that plant is homozygotic dominant because it's making red flowers. We can mate it with a white flowering plant. We know that plant is homozygotic recessive because it's making white flowers, and all of their progeny flowers would be pink because all of those progeny flowers are heterozygotic. So kind of a classic cross here, a Mendelian cross, test cross in fact, that shows that incomplete dominance. So again, the heterozygote has an intermediate phenotype between being homozygotic dominant and homozygotic recessive. To make that red pigment, you actually need the combined work, the combined effort of two dominant alleles, both pumping out this red dye. If you only have one functional allele, you only make half as much of that red dye, that red pigment, and so you fall short of making red and you make light red instead, or pink. So let's predict, if we would, what would happen if you cross two pink flowers, kind of a classic heterozygote by heterozygote cross. Well, if we have two pink flowers, we can kind of imagine this as a Punnett square. We've got one flower with a dominant red allele and one flower with a recessive white allele. The other flower has the same. Again, they're both pink parent flowers. And when we mate them, we see that 1/4 of their progeny is homozygotic dominant, right here, red flowering. Half of their progeny is heterozygotic, pink flowering, and 1/4 of the progeny is homozygotic recessive, white flowering. So our typical 1:2:1 ratio of a heterozygotic cross, 1 part or 1/4 homozygotic dominant, 2/4 or half heterozygotic, and 1 part or 1/4 homozygotic recessive. But what's different is that this is not only the genotypic ratio of this cross, it's the phenotypic ratio as well. If this was a typical gene with complete dominance, we would see this 1:2:1 ratio by crossing heterozygotes. We'd have 1/4 being homozygotic dominant, half of them being heterozygotic, and 1/4 being homozygotic recessive. That would be the genotypic ratio. But the phenotypic ratio would be 3:1. 3/4 dominant phenotypically and 1/4 recessive. If we go back to that idea of brown hair and blonde hair, if we mated two brown-haired heterozygotes, we would expect 75% of their offspring to have brown hair and 25% to have blonde. Here we're seeing something different. In fact, what we see here, and this is a classic hallmark outcome of incomplete dominance, is that the genotypic and phenotypic ratios are exactly the same. What we see in the genotypes is what we see in the phenotypic ratios because each phenotype has its own genotype. Or actually, maybe I said that a little backwards. Each genotype has its own unique phenotype. The homozygotic dominants, the heterozygotes, and the homozygotic recessives all look different. And so we see each of those genotypes manifest as a unique phenotype. So that's incomplete dominance. We also have codominance, which is different. If incomplete dominance is when the phenotype of the heterozygote is somewhere in between, it's intermediate, it's not red, it's not white, it's pink. If that's what incomplete dominance is, then codominance is when the heterozygote has both phenotypes simultaneously. What do we mean by that? Well, we can use blood groups as an example of this. Everybody's basically familiar with the ABO blood group. Most of us know our ABO blood type. I'm O positive, for example. Some of you may be A, B, AB. You might be O like me as well. So we know about that. But there's actually another blood typing system that's rarely important. It's not so medically important, so we don't hear about it much, but it's the MN blood type. Same idea. Some people are M, some people are N, and some people are MN. There's no O in this system. But MN blood typing is a great way to illustrate codominance. Homozygotic MM individuals, so individuals that have two M alleles for this trait, they have M-type blood. And not surprisingly, individuals who are homozygous for the N allele have N-type blood. But heterozygotes who have one M allele and one N allele, they have MN blood. Now, this isn't an intermediate phenotype like the pink flowers, right? It's not that the heterozygote is somewhere between M and N. No. People who are homozygotic for M just have M proteins on their blood cells. People that are homozygotic for N just have N proteins on their blood cells. And people that are heterozygotic MN have both proteins on their blood cells, M and N. So again, the heterozygotes show both phenotypes simultaneously. That's codominance. It's that straightforward. So be sure, as you know, I don't want to think about the exam too early, but we always want to have in the back of our mind the assessments that we'll be doing in this class. You want to have a very clear understanding of the differences between incomplete dominance and codominance, because the differences come down to what happens with those heterozygotes. And if that wasn't enough terminology, we also have incomplete penetrance, which has nothing to do with the heterozygote. Very, very interesting story here. It has to do with how tightly the phenotype correlates to the genotype. So what we just talked about, again, is kind of the basics of refuting Mendel's principle of dominance. That's it for the weirdo dominance things. But we do have other things that would make Mendel say, Imagine two individuals, they're both heterozygotes, they're big P, little p, dominant P, recessive P, and this is for the polydactyl gene. Polydactylism is when you have an extra finger, a sixth finger. It's almost like an extra pinky. That's a polydactylism, and this is the gene that controls that trait. So imagine we have two individuals that are heterozygotic for this trait. They have a dominant allele and a recessive allele. Here's one of those individuals. They have the polydactylism. You can see there's a thumb here, and then we've got a pointer finger, middle finger, ring finger, pinky, and then another pinky, a sixth finger. So polydactylism. So that's one of those heterozygotes, but when we look at the other heterozygote, they don't have that extra finger. They've only got 5 fingers. They have I hate the word normal. I really don't think normal exists, but they have what we would call a normal hand, an average hand with 5 fingers. So what gives? I mean, Mendel says trait equals phenotype. Genotype gives phenotype. That's Mendelian dominance. That's the principle of Mendelian dominance. I don't know if polydactylism is dominant or recessive yet, but I know I'm looking at one heterozygote with that particular genotype and they have an extra finger. That suggests dominance. And then I look at another heterozygote who has the same exact alleles and they don't have the extra finger. So what's going on? Well, it turns out polydactylism is a dominant condition. You only need one big P allele to show the trait. In fact, normal, again, I'm doing air quotes with my fingers because I hate the word, but normal, 5 fingers, is recessive, believe it or not. But only 90% of individuals with the dominant allele have polydactylism. The other 10% have just 5 fingers. This is called incomplete penetrance, when the genotype does not correlate with the phenotype 100% of the time. In other words, when the genotype is not entirely predictive of the phenotype. And this is a big thing that would make Mendel say, what? Because Mendel is all about a direct correlation between genotype and phenotype. So many things can cause incomplete penetrance. Oftentimes it's environmental, and we'll have another example of environmental effects in just a moment. So lots of times when we see incomplete penetrance, it's due to environmental inputs. Different environments manifest phenotypes in different ways. But it can also be due to interactions between genes, and we'll see that in a little bit later in this video as well. Believe it or not, many of you have already observed the effects of incomplete penetrance even if you haven't known it. If you're a Nat Geo fan, a National Geographic Channel or Discovery Channel, if you love animal programs, you've probably seen incomplete penetrance in action and not even realized it. So this animal exhibits incomplete penetrance. This is an Arctic hare. This is a rabbit, or hare to be more specific, that lives in cold environments. And you can see it's a white rabbit, a white Arctic hare, with black tips, just on the tips of its ears. It's got black tips. Some Arctic animals have coloration like this, not only because of their genes, but also because of an input from the environment. So if you take that same Arctic hare that you're looking at there on the right, and if we were to raise it in a much, much colder environment, we would see those little black tips just on the bit of the ear spread. The entire ear would be black, the paws would be black, the snout around the nose would be black. But if we take that same exact bunny, I mean like same exact bunny, genetic clone of that bunny, and we raise it at a higher temperature, we would see an all-white hare. Not even the tip that we see in the actual photograph on the right, we would see a completely white rabbit. So temperature controls the manifestation of this trait. Lower temperature is going to give a rabbit with more black tips, right? And a higher temperature is going to give a white rabbit. So in these cases, obviously fur color is changing with the temperature. This allows the animal to change its colors with the seasons. So in the summer, it's all white. And in the winter, it's got these darker tips. And that helps them camouflage in their environment. This also explains why cold-reared animals have different color patterns near their paws, their ear tips, and their snout. In fact, not to get too personal, but I share personal details quite a bit here. I have a cat. Jet is his name. We've had him now for about 7 years. He's the boss of the house for sure. But when we got him, the day that we got him, I took one look at him and I said to my kids, who were quite young at the time, I know why he looks this way. I know exactly the genetics of why he looks this way. They were so young, they didn't even know what the heck I was talking about. And I was totally geeking out. I mean, even if they knew, I would be embarrassed of myself looking back because I was super excited about the geekiest science thing in the world. But what I was looking at in Jet was that his ears were a dark, dark brown. Right around his nose was dark brown. His paws and his tail were dark brown. His body was kind of more like a light brown, and his belly was almost beige. And looking at him, I knew that the warmest part of his body was the lightest color, and the coolest part of his bodies were the darkest color. Sure enough, came home, looked up the kind of cat he is, looked up that coloration pattern, and it's absolutely incomplete penetrance. The same exact thing as the Arctic hare. So for those of you that have pets, dogs, and cats, you notice these traits in them, it's most likely a temperature effect in their coloration as well. All right, so now we're going to move on to genes interacting with each other. Incomplete dominance, incomplete penetrance, codominance, these are all things that are either nuances in the genetics of how the alleles interact with each other, or nuances in the phenotype and how the environment interacts with the alleles. But everything is one gene. Everything we talked about is the manifestation of one gene. Epistasis is when multiple genes begin to interact with each other to control a single trait. So it's not one gene, one trait, but two genes, one trait. So epistasis is when the genotype of one gene actually controls the expression of a second gene. And that's in fact what epistasis means. The Latin origin of the word epistasis means more powerful than, or stands on top of. So we have an epistatic, so we have this beautiful, cute little cat thing of an epistatic gene and a more diminutive gene. Obviously this guy on the right, he's standing on top of the other. He's more powerful. He's the epistatic gene, or she. So again, one gene controls or dominates or masks another. And that dominance gene, now, and I don't mean Mendelian dominance, right? We're not talking about alleles. We're talking about genes. That more powerful gene, that gene that can stand on top of another, is referred to as the epistatic gene. And the more submissive gene, the one that is being controlled, is the hypostatic gene. So now we can name these cute kitties. This is the epistatic gene, and this is the hypostatic gene. So epistasis, again, one gene controlling another. They don't need to be close to each other in physical space. They're interacting genetically, not physically. And there are two categories or flavors of epistasis. One is recessive epistasis, and the other is dominant, and we'll talk about both. Our example for recessive epistasis will be Labrador Retriever color. So we all know we've got chocolate labs, right? You've got black labs, chocolate labs, and we've got yellow labs as well. So we've got three main coat colors of Labrador Retrievers. Sometimes we call the yellow dogs golden, like golden retriever is actually a yellow dog, but we got black, brown, and yellow. This coat color is controlled by two genes. Now, single trait, right? One trait, coat color. Now, there's three phenotypes in that trait, black coat, brown coat, yellow coat, but that's one trait, coat color. But we've got two genes influencing or controlling that trait. They're in two different locations in the genome. One of those genes determines which color is made for the hair, black or brown, and the other determines how well that color can be deposited in the hair shaft. So we're kind of going back to our example from earlier, brown hair and blonde hair, and in fact a yellow dog or a golden dog is just a blonde dog. We just don't use the word blonde for dogs, and we don't use the word golden for people. So same idea of brown and blonde, but now we've got two genes doing two different things. One gene is making the color, and the other gene is depositing the color. That's really all you need to know in order to understand recessive epistasis. So let's play it out logically. The dominant B allele makes black pigment. So if you have a dominant allele, you as a dog can make black pigmentation for your coat. So homozygous dominant makes black pigmentation for the coat. Heterozygotic makes black pigmentation for the coat. The recessive trait is brown. So only dogs with two recessive B alleles make brown pigmentation. But that's just making the pigmentation. The E gene controls the deposition of that pigmentation. So if you're dominant at E, you can either be homozygotic dominant or heterozygotic, but if you're dominant at E, you deposit that coat color. But if you're homozygotic for E, you get no deposition of that coat color at all. That's a blonde dog, right? That's no pigmentation in the hair. So if you're recessive at E, you are a golden retriever, period. I don't care what your B gene is doing, you are a golden retriever because you can't get that pigmentation into the hair follicles. If you're dominant at E, I still don't know what color dog you are, right? All I know if you're dominant at E is that you can deposit color. The next question becomes, well, what's the B gene saying? If E is dominant and B is dominant, you got a black dog because now you can deposit that black pigmentation. If E is dominant and B is recessive, now you got a brown dog because you can deposit that brown pigmentation. But if E is recessive, period, and it doesn't matter what B is doing, if E is recessive, you're looking at a golden retriever because none of that pigmentation is being deposited in the hair. So I got a little bit ahead of myself there. But to put it in notation, if you're dominant at B, you can make black pigmentation, and if you're dominant at E, you can deposit that black pigmentation. You got this beautiful black dog. If you're recessive at B, you make brown pigmentation, but if you're dominant at E, you can deposit that pigmentation, and you got this doofy-looking brown dog. However, it doesn't matter what you're doing at the B gene. If you're recessive at E, none of that pigmentation is getting into the hair, and you've got this beautiful, and my favorite of the three, I have to admit, this beautiful golden dog. Now again, that dog is golden whether it's dominant B recessive E or recessive B recessive E, because none of that pigmentation is getting into the hair. So that's recessive epistasis. Now, what I want to point out about recessive epistasis is why we give it that name, right? We know that E is the epistatic gene here. E is controlling B. The way I like to think about this is B almost has to ask E for permission. Here's B, I'm B, right? I'm the B gene, I'm dominant, I make black pigmentation, and now I gotta go to E and say, hey E, can I put this black pigmentation in the hair, please? And if E is dominant, it says, sure, go ahead, put that black pigmentation in the hair, make that dog black. And if E is recessive, E says, no dice, I'm in control, I'm in power, that black dye is not going anywhere near the hair, that dog's gonna be golden. So E controls B. B has to ask E for permission. Textbook definition here is the presence of a recessive genotype, little e, at the second locus masks the expression of the black or brown alleles at the first locus. Overcomplication, but saying the same thing, right? If you're recessive at E, you're not getting that pigmentation in the hair, that dog's going to be golden. That's the idea of epistasis. The reason why this is recessive epistasis is because it is the recessive state of E that does the controlling. Well, let me tell you what I mean. So if I were to come to you and say, what form or state of E allows it to say no to B? You'd come back and say the recessive state of E has it say no to B, right? Let's go back to what I said before. I'm B and you're E. I come to you, I've got a dominant B allele, and I say, hey E, I want to make this dog black. Can I make this dog black? You're E. If you're dominant, you say, sure, go ahead, make the dog black. And the dog gets black. I'm dominant B. I want to make the dog black. I come to you and say, hey, E, Can I make this dog black? If you're homozygotic recessive, your answer is no. That dog is going to be blonde. No deposition. So when you, as the epistatic gene, say no to me, you are in your recessive state. And that's why this is recessive epistasis. So recessive epistasis is when one gene controls another, and it controls that gene when it is recessive. No surprise, perhaps, that dominant epistasis is then when one gene controls another when it's dominant. Now one gene comes to the other and says, hey, gene, I would love to make this plant tall. Can I make this plant tall? And the controlling epistatic gene says, no, I control the trait when it's dominant. That's dominant epistasis. So same idea, one gene controlling or suppressing another, but the state of that suppression relies on dominance here rather than recessivity. So in other words, you only need one dominant allele for the suppression. So let's use summer squash as our example for this. Same idea, three phenotypes. You got yellow squash, white squash, and green squash, or zucchini. If you cross a true breeding white squash plant, so true breeding meaning homozygous, with a true breeding green squash plant, all of the progeny in the F1 are white, strongly suggesting that white is dominant, right? Seems like there's two dominant alleles in these white squash, two recessive alleles in these green squash. You mate them and you get all heterozygotes and everyone is white. Now, when you go to the F2, you think you're mating two heterozygotes, and you would expect 3 white squash and 1 green, right? That 3-to-1 ratio. So you do it. You do the self-cross, and wow, what do you see? 12/16 of the squash are white, 1/16 are green. We were expecting 1/4. And 3/16 are yellow. Where the hell did the yellow squash come from? There were no yellow squash in this whole family before. So what's going on? It actually is a 3-to-1 ratio. So the Mendelian genetics does shine through. But the 3-to-1 ratio, if you look real closely, it's 3/4 white and 1/4 color. Right? See that? If you reduce 12/16 into fourths, 12/16 reduces down to 3/4. So 3/4 are white, which is what we predicted. We said we would expect the 3-to-1 ratio, 3 white to 1 green. Well, we've got the 3 white, we've got the 3/4 white, but the 1/4 is not green, it's 1/4 color. So that means a second gene has to be in play. So not making color is dominant. We were right up here when we thought being white is dominant. What it's dominant to is not green, it's dominant to having a color. There's a second gene in play that is controlling which color the squash can be. So when we do the dihybrid cross here, we have our typical 9:3 to 3-to-1 ratio. This is a dihybrid cross of heterozygotes, right? So we have a plant that is big W, big Y, little y, mated with a plant that's big W, big Y, little y. That's our 2 heterozygote dihybrid cross. And we got our typical 9:3 to 3-to-1 ratio. 9/16 are dominant at both genes. 3/16 are dominant at W and recessive at Y. 3/16 are recessive at W and dominant at Y, and 1/16 is recessive at both. But what we see is that both the 9 and the 3 here, 16ths, are giving white squash. It's this 12 that gives us this 12/16 white here. And then we have 3/16 yellow, well that matches what we observed, and 1/16 green, that matches what we observed. So it looks to me like if you're dominant at W, you don't get to make color, right? Because that's the only thing that that 9 and that 3 have in common. Dominant at W, white. Dominant at W, white. Looks like if you're recessive at W, then yellow can be dominant. If you're recessive at W, then green can be recessive. So remember, not having color is dominant to color. But once color can show, then you're off to the second gene to determine the ratios of which color you're observing. So gene W is the epistatic gene. Gene Y is the hypostatic gene. Gene W is in control. But unlike the Labrador story from the previous slides, when Y comes up to W and Y says, hey W, I'm dominant. I'd love to make this squash yellow. Please, can I make this squash yellow? W says, sure, when it's recessive, giving these 3/16. But if W is dominant, W says no, no color for you, white squash only. Or if Y is recessive, it comes over to W and says, W, I would love to make this squash green, please. And if W is recessive, it says, sure, go ahead, make that 1/16 green. But if Y is dominant, it says, if W is dominant, it says no, no color for you, it's going to be a white squash. So not only is W epistatic, not only is W controlling the trait, but W controls the trait when it is dominant. Hence this being dominant epistasis. W says no to Y when it is dominant, making this dominant epistasis. So only when W is recessive does the Y gene get to express. If W is dominant, the Y gene is masked. If dominance is saying no, then this is dominant epistasis. And just on the back end here, I just think it's super interesting and remarkable that we do see the 9:3 to 3:1 ratio here. This is classic Mendelian genetics. Remember, everything in this lecture are things that would initially make Mendel scratch his head and say, what? But upon deeper digging, if you truly understand the genetics, you can see why these things are behaving this way. This 12:3:1 ratio is really a 9:3 to 3:1 ratio masked, hidden in the phenotypes. So it is following typical Mendelian genetics. Sometimes we just have to look a little harder to see it. So we've got our two examples here, right? If we've got no epistasis in play at all, then we're going to see our typical 9:3 to 3:1 ratio. There's no interaction between these genes. We have two distinct phenotypes, right? Maybe it's seed shape and seed color. So 9 out of those 16 seeds would have the dominant shape and color. 3/16 would have the dominant shape but the recessive color. 3/16 would have the recessive shape but the dominant color. And 1/16 would be recessive for both shape and color. Typical, standard, 2 genes, 2 traits, dihybrid cross, 9:3 to 3:1. If we have recessive epistasis, like we saw in the Labradors, then we have no. The epistatic gene is saying no when it's recessive. So these two groups cluster. And what we see is black dog, brown dog, golden dog, a 9:3 to 4 ratio, black to brown to golden, because the epistasis is coming into play, and the answer is no when the epistatic gene is recessive. In the summer squash example, we have dominant epistasis. Now we're clustering these two genotypes, because when the epistatic gene is dominant, it says no. So we've got white squash, yellow squash, green squash, a 12:3:1 ratio. But again, it's all the 9:3 to 3:1 hiding in these clustering or combining of genotypes and phenotypes. All right, another example of two genes interacting, far less complicated than epistasis, but an interesting story nonetheless, is complementation. By integrating everything we've just learned about, namely that two genes can interact with one another, we can actually do a neat little genetic trick. We can determine if two different mutations in two different individuals for a single trait are on one gene or on different genes. And we do that through complementation. So here's what I mean, for example. In this case, we've got two genes, gene A and gene B, and let's assume, for argument's sake, that both gene A and gene B control a single trait, like coat color in a dog or like squash color. In this case, the individual has a mutation in their A alleles. In other words, they're homozygotic recessive for A, and they're homozygotic dominant for B. So they've got mutations in both of those genes. In this example, we've got a mutation in one gene, the A gene, in one individual, and we have a mutation in the other gene, the B gene, in a second individual. So here the two individuals both have mutations in their A gene, and here one individual has a mutation in the A gene and the other has it in the B gene. And by using complementation, we can distinguish between these two, and we can figure out which is actually occurring. So in this example, let's just assume it's for brown eyes. Let's say that gene A and gene B work together to make eyes brown. And in order to have brown eyes, you have to be dominant at both genes. You can be homozygotic dominant or heterozygotic, but if you're dominant at A and dominant at B, you've got brown eyes. And if I haven't pointed it out yet in class, these underscores are really just placeholders. They're meant to suggest that there is another allele here, but it doesn't matter what it is. If this is a dominant A or a recessive A, this is a dominant B or recessive B, it doesn't matter, because all you need is one dominant A and one dominant B to have brown eyes. If you're recessive at A and dominant at B, or vice versa, dominant at A or recessive at B, or if you're recessive at both, you've got blue eyes in this example. So in other words, the only way you can have brown eyes is being dominant at both genes. If you're recessive at either gene or both genes, you're going to have blue eyes. So we take two blue-eyed mating parents and we mate them. And we're asking, are the mutations in the same gene, or is there complementation going on? In other words, does more than one gene control this trait? So let's consider the same gene first, right? Typical classic Mendelian genetics. Parent number 1 is blue-eyed because they have a mutation in their 1A gene. They're homozygotic recessive at A. And in this case, they can be dominant at B. It doesn't really matter. Parent number 2 is blue-eyed, also recessive, because they also are homozygotic recessive at A. So same genotype for all intents and purposes. Again, same mutation, same gene, homozygotic recessive at A for both. And they mate. Is it possible for these two parents to ever have a brown-eyed child? And the answer is no, because all either parent has is recessive A alleles. Every single offspring between these two parents is going to be homozygotic recessive for A. There's no dominant A allele in this mating anywhere. Those kids are all going to be homozygotic recessive for A, and they're all going to have blue eyes. So no, they can never have a brown-eyed child. Let's consider the same exact phenotypes in the parents, but now those mutations are in different genes. Let's say that parent number 1 is blue-eyed because, again, they're recessive at A. But let's say that parent number 2 is blue-eyed because they have a homozygotic recessive genotype at B. And we'll ask the same question. Is it possible for these two parents to ever have a brown-eyed child? And the answer is yes. Because those mutations are on different genes, these parents can have a child that is heterozygotic for A, heterozygotic for B, and remember, all you need for brown eyes is one dominant allele for each gene, and you'll have brown eyes. This would be a brown-eyed kid. So if this dominant A went into a gamete with this recessive B, and if this recessive A went into a gamete with this dominant B, and those two gametes fused, you would have a child that's dominant recessive for A, dominant recessive for B, heterozygotic for both genes, and that's a brown-eyed kid. So when that happens, when you can get a wild-type dominant trait like brown eyes by two mating recessive parents who each had blue eyes, the only genetic explanation for that is complementation. In other words, those two genes are different. The mutations are on two different genes. So again, we'll give another example here, more biochemical example, purple pigmentation in flowers. So this is meant to show a very crude and rudimentary biochemical pathway. We have some colorless compound or precursor number 1, and gene A encodes an enzyme that converts number 1 into a colorless compound number 2. And gene B encodes an enzyme that converts number 2 into a purple compound that makes the flower purple. And it's dominance at A and B that encode the working enzyme. Recessivity at either of these genes is a lack of function condition, and that means the allele doesn't work. The enzyme doesn't work, and you can't get all the way to purple. So for example, if you have this genotype, homozygous dominant at A, you're making functional A enzyme, so you can convert colorless compound number 1 into number 2, but because you're recessive at B, you don't make any functional B enzyme, and you can't turn number 2 into purple. You're going to have a white flower. And likewise, if you are homozygotic recessive for A, you can't make functional A enzyme. You'll never convert colorless compound number 1 into number 2. Now, you've got a functional B enzyme, so if you had 2 around, you could make it purple, but there ain't no 2 around because you can't ever get out of compound number 1. So the only way to be purple is to be dominant at A and dominant at B. So imagine the cross between these two different white flowers. Imagine we take this white flowering plant that is homozygotic dominant for A and homozygotic recessive for B, and we mate it with this white flowering plant, which is homozygotic recessive for A and homozygotic dominant for B. If we imagine those individual Punnett squares in our head, we see that this parent can only give dominant A alleles. This parent can only give recessive A alleles, so every single child is going to be heterozygotic for A. This parent can only give recessive B alleles, but this parent can only give dominant B alleles, so every single child is going to be heterozygotic for B. And if you're heterozygotic for A, you're making good A enzyme. You're going from 1 to 2. If you're heterozygotic for B, you're making good B enzyme. You're going from 2 to purple. You're going to have all purple flowers, all purple offspring. This is yet another thing that would make Mendel say, what just happened? In Mendel's world, recessive mated by recessive always gives recessive. Yet here we're seeing recessive mated by recessive gives wild-type dominant. Mendel wouldn't understand this at first blush, but as we dig deeper into the genetics and what we understand is that two genes control the trait and complementation is occurring. In other words, what's broken in one plant is fixed by the other. What's broken in this parent is fixed by this one. That's complementation. We can get back to a wild-type phenotype. All right, so we'll talk about sex-influenced and limited characteristics and then environmental effects, and we'll be done with this lecture. So obviously, sex-influenced means that the sex of the animal, the sex of the individual, is going to influence or control something about the trait. So these are traits that are genetically inherited, and they are influenced by the sex of the individual. However, these are not traits that are encoded on the Y or X chromosome. You may have learned about X-linked traits like colorblindness and hemophilia in other classes. Those are sex-linked traits. Those not sex-influenced or sex-limited. These are traits that are on autosomal chromosomes, non-sex chromosomes, but are still influenced by the sex of the individual. So how is a trait on an autosomal chromosome sex-influenced? Oftentimes we don't really understand why, but we can still observe the effects. Sex-influenced traits are defined as traits that are determined by an autosomal gene, a gene on an autosomal chromosome, that can be inherited following Mendel's basic principles, but expressed differently in males versus females. To say it more simply, the trait is dominant in one sex and recessive in the other. Sex-influenced traits can be expressed by both sexes, but the penetrance is different because of this kind of flip-flopping dominance recessivity. And we already know what penetrance means in genetics. We mean the correlation between a genotype and a phenotype. So in other words, to say it simply, one sex expresses the trait more easily than the other. So let's use billy goat beards as our example, right? Goats can have beards. We know that. We see, oh my goodness, how did he get there? That's so unflattering. But anyway, we see these goats here. They have these beards. And I guess we could say that this is the same kind of beard here as well. Typically, a billy goat or a male goat has the beard, but female goats can have beards too. It's more correct to say that male goats are more likely to have a beard than female goats, and we understand why. It's because this is a sex-influenced trait. It has a different penetrance in each sex. So the allele that causes beards in goats is technically a recessive allele. It's this kind of big B superscript little b. And the allele that gives no beard is this B plus allele, technically dominant. But here's the sex-influenced part. That bearded allele is actually genetically dominant in males. In other words, if you're a male goat, you only need one of these BB alleles to have a beard. That's Mendel's definition of dominance, right? Whatever phenotype you show with only one allele is the dominant trait. So all you need is one BB allele to have a beard in males. But if you're a female goat, you need two of those alleles to have a beard. So the implications of this is that you have more male goats with beards than females. There are only 3 possible genotypes for this trait. It's a single gene trait. So you can have plus genotypes, you're heterozygous for B+. You can have BB genotypes, you're heterozygous for BB. Or you can be heterozygotic, where you have one of each allele. If you're a male, you are beardless only with the plus genotype. Because again, if you're a male goat, you have the beard with one B allele. So heterozygotes have a beard as a male, homozygotes have a beard as a male. If you're a female, the only way you can have a beard is by having 2 of these alleles. So here the heterozygotes are beardless, so much fewer female goats have a beard. So if you have 2 heterozygotic goats, now here's where it gets interesting, right? You have 2 heterozygotic goats of different sexes. Based on that, the male goat has the beard, and the female goat does not have the beard, right? Because that's the sex-influenced effect. Incomplete penetrance. When they mate, 75% of their male offspring would have beards. Those would be the male offspring who are heterozygotic, and the male offspring who are homozygotic for this trait. this allele. The other 25% of the males would not have beards. They would have this genotype. However, only 25% of the females of that same exact mating would have beards. That would be these females with this genotype. In the females, these would be beardless females, and the heterozygotes would be beardless females. So again, sex-influenced. Same exact genotype, different penetrance of the phenotype based on sex. The reason why we call this sex-influenced, though, is because, again, both sexes can show the trait. We can have bearded males, we can have bearded females, we can have beardless males, we can have beardless females. All that differs is what the heterozygote does. What's the appearance of the heterozygote? Sex-influenced. And we've got some schematics here as well. This is basically diagramming the cross we just said. So here's your beardless male. He's ++. Your bearded female is BB. They give rise to nothing but heterozygotic goats. Even though all those progeny goats have the same exact genotype, heterozygotic, all the females are going to be without a beard, and all the males will have a beard. And then we can do that heterozygote by heterozygote cross. There's no real surprise here. We've got a 1 to 2 to 1 ratio, ++ to heterozygote to BB. But where it gets weird is what the different sexes look like. 3/4 of the males have a beard, 1/4 do not. 3/4 of the females do not have a beard, 1/4 do. So that's sex influence. Sex limited is similar. It's really just an extreme form of sex influence. In a sex limited trait, one sex cannot show the trait, period. Doesn't matter what the alleles are doing. The trait can only be shown in one of the two sexes. So here, only one sex ever expresses the trait. We'll use rooster feathering as our example for this trait, and keep in mind that these traits are governed by autosomal genes. Again, none of these genes that we're discussing are on the X or Y chromosome. I'm not talking about sex-linked traits at all in this class, because I'm assuming you've had that already. No color blindness, no hemophilia. This is all autosomal traits. But again, for sex-limited traits, there is zero penetrance in the non-expressing sex. Some roosters, not all, but some roosters, again, a rooster is a male chicken. Some roosters express the cock feathering trait. That's this kind of typical foghorn leghorn rooster that you see. The beautiful plumage on the rear end of the rooster. Some male roosters have this and some don't. But hens never have this. Female chickens never have this cock feathering trait. The other phenotype expressed by some males and all females is called hen feathering. And again, it looks like this. I'm no chicken expert. If I saw a chicken like this in the wild, I would have no idea what its sex was. This could be a male chicken with hen feathering or a female chicken with hen feathering. However, conversely, if I saw a chicken that looks like this on the farm, I would immediately know that must be a male chicken, a rooster, because only male roosters can have this trait, this plumage on their rear end. So cock feathering is recessive, but it is sex limited. It will only express in males. We can have homozygotic dominant males, and they will have hen feathering. Homozygotic dominant females will have hen feathering. Heterozygotic males will have hen feathering. Heterozygotic females will have hen feathering. Homozygotic recessive males will have that cock feathering plumage, but homozygotic recessive females will still have hen feathering. Again, sex limited. And we're not sure the physiological basis of this. Finally now, environmental effects on phenotype. We've already talked quite a bit about nature and nurture, whether we realized it or not. Anytime the environment has an influence on a trait, we're talking about this nature-nurture divide. Nature being the genetics and nurture being the environment. So the Arctic hare, for example, and my cat Jet, is a merging of nature-nurture and incomplete penetrance. These concepts address the role played by the environment in controlling phenotypes, or at least influencing them. But we can talk a little bit more about the role played by the environment and how it gives about exceptions to Mendel's rules. Mendel states that two individuals with the same exact genotype should be identical in their phenotype, and we've already seen that's not necessarily true. Textbooks will say the genotype sets the potential for development, but the phenotype develops within the confines imposed by the environment. And this is very, very normal, whether we realize it or not. Let's think about human height, for example. Now, human height is controlled by dozens of genes, but it is genetic. So presumably, if we had the technology, we can screen a human being's genome for all of the height governing genes and determine if this individual should be very tall or very short. And let's imagine, fast forward maybe 15 or 20 years, and let's imagine we can do that, and we have a 2-year-old with the predetermined genes for being very, very tall. So maybe we can fast track that kid into basketball practices and basketball clinics. We know they're going to be really tall, right? There's no incomplete penetrance of height. There's no multi-gene interactions of height. This kid's got the alleles for being very tall. We come back 10 years later and the kid's short. And we have no idea why. We dig a little deeper and we realize that the kid was actually raised in a socioeconomically challenged home. And oftentimes there was not enough food to eat. This kid was malnourished for much of those formative years between toddler and adolescence. And as a result of that malnourishing, that kid grew to be much, much shorter than they were genetically predetermined to be. So that's nature-nurture. That is actually incomplete penetrance. We would always say that height is not incomplete penetrant. Height is governed by genes, but the environment still has a role to play because in order to grow to our maximum potential, we need proper nutrition. So again, the genotype sets the potential for the development of a trait, but the phenotype develops within the confines imposed by the environment, in that case, nutrition. We'll use vestigial wings here as our example. So that's a mutant or a recessive phenotype. VG is the mutant alleles. These flies will have vestigial wings, very, very small curled wings that don't allow the fly to fly. But this only happens when the fly is reared at colder temperatures, temperatures below 29 degrees centigrade. If we rear those same exact mutant flies at a higher temperature, they will actually have normal wings, even though they're homozygotic recessive, even though they're mutant. So what's going on here? This is obviously an environmental effect, much like the Arctic hare. Temperature seems to be playing a role, but this temperature is playing a role between a mutant phenotype and a wild-type phenotype. PKU, or phenylketonuria, is another example of the same exact thing. This is a genetic disorder that can cause mental and cognitive defects, but only when people are bombarded metabolically with phenylalanine. So this genetic mutation mutates an enzyme that is needed in order to metabolize the amino acid phenylalanine. Individuals with phenylketonuria cannot metabolize this phenylalanine amino acid, and it builds up in their bloodstream and causes toxic effects. It's the toxic buildup of phenylalanine that actually manifests the symptoms of the disease. We can diagnose PKU at birth with a genetic test. We can identify newborn babies that have PKU, and as soon as they're identified, they can be put on a very special low phenylalanine diet. If we decrease the amount of phenylalanine that these babies and ultimately adults consume, they'll never manifest the symptoms of the disease because they never have a toxic buildup of phenylalanine in their body. So by manipulating the environment, we can manipulate the phenotype. We can spare these individuals the disease of PKU by keeping phenylalanine out of their body, by keeping it out of their diet. And you might notice sometimes, especially on soda cans with artificial sweeteners, they'll have a note there by the ingredients that says, "Attention phenylketonuria, this contains phenylalanine." That's a warning for PKU individuals not to consume that, because that could lead to a toxic buildup of phenylalanine. And here's some examples of that kind of warning label. Sometimes the environment alone can cause a nearly identical trait to a particular genotype. Back to flies, there is a genetic mutation, a genetic phenotype called eyeless, which causes very, very small eyes to form in a homozygotic recessive individual. This is a normal wild-type fly with big eyes. This is an eyeless fly, a genetic eyeless fly. But we can take wild-type flies and expose them to a compound, a chemical, sodium metaborate, and it causes the same exact phenotype. This is called phenocopy, where we can mimic or copy a phenotype that is typically genetic with the presence of a chemical. Usually the chemical is disrupting the enzyme that gene coded for in the mutant. So we can kind of deplete the cells of the working enzyme by impeding its action with a chemical, just like the mutant alleles impede the action of the enzyme by not coding for it properly. All right, so that's all I have prepared for you here. This is our first introduction into things that would make Mendel say, "What?" apparent contradictions to Mendel's rules that are not contradictions at all as we dig a little deeper. We talked about incomplete dominance. Remember, incomplete dominance is where the heterozygote is intermediate. We've got red flowers being homozygotic dominant, white flowers being homozygotic recessive, but the heterozygote is intermediate, light red or pink. We've got codominance when the heterozygote has both phenotypes simultaneously. Our example there was MN blood types. Incomplete penetrance is where there's not a direct 100% correlation between a specific genotype and a phenotype. Our example there was the heterozygote for polydactylism. Some people, about 90% of people who are heterozygotic for polydactylism have an extra finger, but 10% don't. And the Arctic hare is another example where the environment is actually driving the incomplete penetrance. Then we moved on to epistasis in both forms, recessive and dominant epistasis. This is where the genotype of one gene can control the expression of another, where the hypostatic gene must ask the epistatic gene for permission to express. And if it's recessive epistasis and the epistatic gene is recessive, the answer will be no. If it's dominant epistasis and the epistatic gene is dominant, the answer will be no. If mutations for the same trait are on different genes, those mutations can be complemented by wild-type dominant alleles in the other parent. So if we take two mutant parents showing a recessive phenotype, and we mate those two recessive parents and we can generate a wild-type dominant offspring, we are looking at complementation. We talked about sex-influenced and sex-limited characteristics. Influenced means both sexes can express the trait, but that trait is dominant in one and recessive in the other. Sex-limited means only one sex can express the trait ever, and the other sex must always not express that trait. And finally, we wrapped up with this idea of the environment interplaying with the genetics, playing a large role in the phenotype, and we can even have phenocopy, where the environment can completely mimic a phenotype even without the presence of any recessive alleles. So, that is your first Edpuzzle lecture for the course. Hopefully you did like it, at least at some points. This will be the flow of the course. You'll watch one of these types of lectures each week. Questions will pop up in the middle of them, as you saw. Please let me know, as the dialog box pops up in just a moment, what is still confusing to you, and we'll take about the first hour of class together when we meet on Friday to review those most complicated topics, and then we can move into challenge questions and paper dissection. So, until I see you again next time, thanks so much for watching, and have a great day.